Dr. G, Dr. Randy Guliuzza, it is great to have you here for our Science Confirms the Bible seminar. You are going to be giving a presentation, Deconstructing the Engine of Evolution. Science shows that animals are pre-engineered to adapt and track their environments. I'm excited for this. I will let the audience know we will be wrapping up the night with an audience Q&A period. So if you hear anything said or presented during any of the talks tonight, feel free to send in your questions or challenges or objections, and we are going to have a Q&A period with the full panel as soon as we're done with the talks. And so, Dr. G, we're going to give you a formal introduction before we give you the floor for your presentation. Sam, would you do us the honors, brother?
I'd love to. So Dr. Randy Guliuzza is the president of the Institute for Creation Research. He is a leading biological design theorist and is actively expanding ICR's scientific research and heading the critical endeavor to develop a theory of biological design. He has represented ICR in several scientific debates at secular universities and other forums. He holds to a Doctor's of Medicine from the University of Minnesota and a Master's of Public Health from Harvard University. Dr. G also has a BS in Engineering from South Dakota School of Mines and Technology and a BA in Theology from the Moody Bible Institute. He was board certified in aerospace medicine, and he is a licensed professional engineer.
Dr. G, how are you tonight?
Oh, I'm doing great. Thank you so very much for this invitation to be here.
Well, it's our pleasure. I'm looking forward to this, and therefore I'm going to get your slides up here. I've got it up on the screen, and Dr. G, the floor is yours. Go ahead.
All right. Well, thank you very much. I hope everybody was listening very carefully to my colleague, Dr. Brian Thomas, there, with especially the gospel presentation that he had right at the end. Now, you probably already noticed immediately that I speak a lot faster than Dr. Thomas, and I really have to kind of move tonight because we have to cover a lot of material. In fact, even this title that I was assigned here is kind of long. So we need to move right back into what we're talking about. And everything I'm going to talk about tonight, almost everything, is already in this paper. So I know people will be watching this later on recording, so they can call up these slides later. But if you're interested in what I'm talking about, this was a paper that was published at Cedarville University at the ICC, International Conference on Creationism, just in 1923. So this will have a lot of the background information that we won't be able to cover in this short period here. But I do want to answer a challenge by some evolutionists, and there's a couple of them on the screen right there. And a lot of times we just push back against evolution, but they have this question a lot. What do you have to offer instead? You know, what do creationists have to offer instead, other than just saying God did it? And of course, we don't say that, but that's the expectation. In fact, an evolutionist gave a really good challenge that I think sets the stage for tonight's talk. He sent this in a text message, and he basically asked, here's my number one question for creationists, and in this case, he's including intelligent design people too. What's your research agenda? That is, what do you do next? And my biggest problem with this worldview, he's talking about ours, is simply that I've not heard of any positive consequences for research. What new research does your hypothesis help you do? And what would you do with a $5 million research grant? You know, that's a good question, and I hope to answer his challenge here tonight by saying that we actually have developed an alternative theory, a theory of biological design. And that's covered in the paper that I referenced at the very beginning, but I do want to cover some of the major points of a theory of biological design and really what a theory is going to help us do, what evolutionary theory helps people to do as well, is answer big questions in a particular field. And then after you do that, interpret what you see. And so evolutionary theory does that for evolutionists. So we need to develop a theory that answers the big questions as well and guide our interpretations. And the major observation in biology that really needs explanation is design. In other words, how do you account for the fact that creatures look so incredibly designed? And we see it, and evolutionists see it. It's the major question that needs to be answered, not only by their major organ systems and things, but also by their behavior, which is highly purposeful as well. And of course, how well they fit their environments, because, birds look like they fit in the air and fish look like they fit in the ocean. How in the world do we explain all of that? Well, Dr. Paul Nelson, he came up with this wonderful, wonderful guide, which is that we need to begin doing biology as if Darwin had not been born. And I wish I would have said that myself, but he's basically calling for a fresh break, not to revise evolutionary theory in any way, but to come up with something completely different than Darwin's theory. And this is a quickie picture from Darwin's Field Book, and it really shows the beginning of a theory. And he begins up there with, I think, and a lot of times creationists and ID people criticize him for that. In other words, he's like thinking or speculating, but that's what a theory is. In many ways, it's a narrative to help guide our thinking. And you see there, he's speculating not just on the diversity of life, but why creatures look so incredibly designed, because every one of those nodes at the end is supposedly a creature that is so incredibly fit to their environment that they have actually developed exquisite design. So he begins by, I think, and that's what a theory is. A theory is a hypothesis. It's usually a working hypothesis that's supposedly a plausible explanation for how something works or how something originated. And Darwin came up with one of those theories, and it's really dominant these days. But what do we have to offer instead? We have a theory of biological design, and so I'm going to lay out some of my assumptions, the things that I think that are very, very contrary to evolutionary theory and specifically related to engineered biology. So I think that the reason why creatures look so incredibly engineered is because they are, in fact, engineered. And because of that, I also hypothesize that engineering principles will explain the operation of biological functions. I don't know of any evolutionists that would hypothesize that, but if I'm going with an engineering-based explanation, I would hypothesize that that's going to happen. In addition, a good theory makes predictions. So I predict under this theory that all biological systems can be reversed engineered. And evolutionists say that creationists don't make predictions, but this is one that would be easy to disprove. Just find one that can't be reverse engineered. In addition, I predict finding goal activity directed at every research level. That's from the organ system all the way down to the organs and the molecular level. I expect to find this activity at every single level. I also predict finding corresponding systems underlying similar engineered and biological functions. That's something that evolutionists wouldn't predict, but from an engineering perspective, I predict finding those as well. I predict finding identifiable control systems in organisms directing all of their operations. That's another thing that I would predict to find. And all of those predictions need to be set into the context of what I'm really going to talk about and what I'm not going to talk about. So in the realm of engineering, I would roll in all of the major biological functions of organisms, which is their growth and reproduction, adaptation, and metabolism. All organisms do those things. And so I am suggesting that in terms of how they operate, they all will be explained by engineering principles. But I am specifically not talking about life or consciousness or agency. And in fact, I don't know of anybody that has actually researched life directly. I don't know of a single scientific paper where anybody has held a beaker of life and done any experiments on life itself or consciousness for that matter. So I am not talking about those things. In fact, I would argue that they're not reducible to engineering principles or to biological functions, that they exist outside of them, that they're immaterial. And this is why humans really cannot create life, and creatures like you and me are not really machines. So I'm talking about the actual biological functions that organisms are doing: growth, reproduction, adaptation, and metabolism. And tonight we're going to look specifically at adaptation. So if we have everything in this context of a theory, where we have some hypotheses of what we're expecting to find and some predictions, then we need, as our first step, is to make new models. And so we want to put together a model of adaptation, an engineering-based model of adaptation that focuses on the organism. And because it's engineering-based, it eliminates all references to mysticism. In other words, nothing that cannot be quantified and actually identified is put into our explanations whatsoever. If you can't quantify it and you can't identify it, we don't put it into a causal explanation. So let's look at a model that explains adaptation without any mysticism, focuses on the organism, and is engineering based. And that model that ICR has developed is called continuous environmental tracking. And other organizations have picked up on this model as well. And it also makes, like any good model, an hypothesis. And the hypothesis is that what creatures seem to be doing is continuously tracking environmental changes. And this is not that they're passive in the slightest, but they are active problem solvers. And they're actively tracking what's happening around them, all the way from the level of their metabolism and homeostasis up to acclimation and even multi-generational changes. And because they're able to purposely track, they're able to have purposeful adaptations. Now, this is very, very different from an evolutionary explanation. Everything is active by the organism. And so that's why we hypothesize that organisms are tracking. And how they're tracking is due to many innate systems that they're actually activating. And therefore, I expect the characteristics of what they're doing to be consistent with the characteristics of design systems. And so there are several distinguishing features of continuous environmental tracking compared to Darwinian selectionism. First, CET is organism focused. It's not focusing on the environment. Anything that any of those creatures you see on the screen can and ever will be able to do is inside of them. Any way they relate to each other or their environment is inside of them. So if you want to figure out how they operate, you focus on the organism, and that's where you'll find your explanations for adaptation. Second, as I've already alluded to, it's an engineering-based explanation. Therefore, from that, we make all of those predictions of what we expect to find and how we expect these organisms to operate and adapt. So one of the engineering-based expectations I have is that their capacity to adapt is built in upfront. So just as the engineers surveyed all the different environments that a space shuttle would need to go to and expected all of those challenges, then they built into that space shuttle the ability to solve those challenges. And therefore, from an engineering perspective, I expect the solutions to precede the challenges, and not from an evolutionary perspective as due to those challenges. So the ability to adapt to many different environments, of course, it's limited, just like any engineered thing. It has parameters that it operates within. But one major distinction is that those are built in up front. And therefore, I expect to find corresponding system elements. So on the left-hand side of the screen, people are looking at an autonomous car that can drive itself around challenges, around storms. And it does so because it has many built-in sensors, innate logic mechanisms, and a response capability to do those things. Therefore, on the right-hand side of the screen, when I'm looking at guinea pigs under this sign that says guinea pigs beat climate change by tweaking their own DNA, I'm expecting to find those same system elements of sensors, innate logic, and this response capacity. And in this case, they took some male guinea pigs, put them in cages with heating pads underneath to simulate climate change, and they kept them there for 70 days, the time it makes to make one cycle of sperm. Then they mated them with female guinea pigs, and they analyzed the pups and the dads. And within that same generation, at least 18 different genes had different expression on them as they related to temperature regulation. And that's how they're able to tweak their own DNA. And so therefore, from a research program agenda for a real theory, you would have one. I'm looking for those engineering principles and those corresponding system elements, the sensors, logic, and response mechanisms in that guinea pig. This is what my research program would have me looking for, and I expect to find them, and I predict that we would find them. Another example is of these rove beetles and how they're able to track their environment. Now, everybody can see the dark brown beetle on the left, and they can see the ant on the right, but that isn't an ant. That is the rove beetle. And that beetle can morph into the shape of an army ant, which is its natural enemy. And nobody has actually seen this happen, but it happens on multiple occasions. And there on your screen, you can see an army ant and you can see a rove beetle, which is morphed to look and smell and behave just like that army ant. And we can track over time that these same beetles have been able to do this for at least 9 different types of army ants. Quite remarkable, quite incredible that they're able to morph into these different shapes. In Current Biology, which reported on this, proposed that the quote, as you can see there on the screen, these beetles were poised for mimecophilia, that is to look like an ant. And this near clade-wide pre-adaptive ground plan may underlie their repeated ant mimicry. And the American Academy of Sciences has Eureka Alert. They reported on this exact same thing, and they report that the discovery published in Current Biology provides evidence that evolution has the capacity to repeat itself in astonishingly predictable ways, completely contrary to evolutionary thinking. Another study in Current Biology reported on stick spiders in Hawaii. And there on the screen, you see a gold one that lives on leaves. But there's a white one which lives on lichen and a brown one which lives on tree trunks. And they found that, say, this gold one went to one of the Hawaiian islands, it can morph into the white and brown forms before the white and brown forms from this preceding island could even get to that island. And these researchers said, we usually don't expect evolution to be predictable, but Hawaiian stick spiders of the Rhamnus genus have repeatedly evolved the same distinctive forms known as ecomorphs on different islands. The spiders arrive on an island and boom, you get independent evolution to the same set of forms, this sort of rapid and repeated evolution. We can ask whether there might be an underlying mechanism that leads to the similar patterns of predictably repeated evolution in the course of adaptive radiation, that the Rhamnus spiders have some sort of pre-programmed switch in their DNA that can be quickly turned on to allow them to evolve rapidly into these successful forms. But how that process might work is still unclear. I suggest that they look for the sensors and the logic mechanisms and the output responses. Another paper reported on predictable evolution trumping randomness of mutations. And this was research on E. coli in separate colonies which were exposed to identical stresses. And they reported that although the mutations, the driver of evolution, occur at random, a study on the bacterium E. coli reveals that nature has can find the same solution to the same problem again and again. Identical mutations appeared independently in the test tubes. And they went on to say that the E. coli bacteria can acquire predictable mutations to adapt in changing environments. And then I have one final study, which was on fruit flies. And there are many, many, many of these studies reported in our paper. And this tracked 10 different populations of over 100,000 flies in each population over a season of just 4 months. And they continuously repeatedly sampled their genome for changes in allele frequency. And they found that all of the populations were essentially tracking with each other as there were major changes in their allele frequency just over that summer period. And the research was done at Penn State, and they reported on that research of the rapid adaptation in the fruit flies that they normally expect to take a long time, but was actually happening very, very rapidly within just a few weeks. And to what extent are we talking about? They report that over the course of the experiment, which lasted 4 months, the researchers documented changes in 60% of the fly's genome. With this direct observation of swift and continuous adaptation in response to the environment, a phenomenon known as adaptive tracking, the biologists have established a new paradigm for how to think about this in new timescales. And one of the major researchers, Dr. Schmidt, mentioned that we see the populations are able to track differences in the environment. The populations were constantly evolving and shifting throughout the experiment. I would argue that these processes are taking place in a lot of different organisms, but they're harder to measure over the appropriate timescales. So we see that these organisms are able to attract their environments in highly regulated, rapid, repeatable, and with very, very targeted responses. All of this is fitting the engineered basis of continuous environmental tracking. It's far more consistent with that than random mutations being fractioned out in deadly struggles to survive. Now, after you develop a model, the next thing you need to do is develop new research in order to test those models in head-to-head tests. And continuous environmental tracking is really the first anti-Darwinian explanation, which is a thoroughly engineering-based description that's completely focused on the organism and focuses on non-random changes there. So what were we able to find? As we looked at these cavefish, which is what we're doing research on at ICR, we noticed that there is, and everybody can see this, there is a change in their pigmentation from the sided fish, which lives in streams, to the hypopigmented fish, which lives in caves. And these are the same species in a sense because they can mate with each other and have fertile offspring. So you ask the question, of course, what leads to the reduction in eyes and what leads to the reduction in their pigmentation? And evolutionists have an explanation for that, which is like all of their explanations. And this paper by Borowski in 2018 mentions that they believe it's due to a loss of function mutation in the OCA2 gene. And then they believe that this is happening due to this, as I mentioned, loss of functions. And for reasons currently unknown, OCA2 seems to be evolution's favorite for pigment regression. Therefore, we can put a head-to-head test their predictions, where they would predict that this loss of pigmentation is due to random and broken genes. And it's overwhelmingly, according to them, a result of broken genetic pathways and due to a loss of function. Well, we would have completely different predictions. We would predict that these fish were engineered up front to live in these caves, and that the changes in pigmentation and many, many other changes that you would see are not due to broken pathways, but are traits that can be downregulated and upregulated in a very purposeful and regulated fashion. So this allows us to put a head-to-head test in a real research program and answer to the evolutionist challenge, which we had right at the beginning of this about 20 minutes ago. And so as we put this to a head-to-head test, we have to ask which one of these provides a better explanation. So here's a fish that we tested. Obviously, it's a blind cavefish, hypopigmented. This is a female in the natural environment which she would be at, actually in a cave. It's in our aquarium right now. But we subjected her to simulated sunlight with the same intensity, wavelength, frequency, durations as if she was now reintroduced back into a stream. And the evolutionary prediction is that her genes are broken, therefore she should stay in this hypopigmented state. Our prediction is that she would regain her pigmentation because it can be upregulated just as it was downregulated. And this is the fish after 32 days of sunlight exposure. Clearly, ability has not been broken. The ability remains there. And this is the male fish exposed at the same time. Both of these are still alive. They've just been stunned a little bit so we can take this picture. And this pigmentation can be regulated up and down as these organisms continuously track environmental changes. Therefore, we have two explanations. The evolutionary random adaptive process, which they say is not designed, which sees passive organisms shaped over time, and their changes are basically undirected, trial and error due to iterative processes, which Darwin said were numerous successive slight modifications, put to a head-to-head test against one which sees organisms as designed, with purposeful adaptive processes, not as passive, but as active problem solvers, and with mechanisms that are highly regulated, producing targeted solutions that are predictable and repeatable, just as I gave by all of these examples. And It is a system that can track the environments in many directions which it can go within its design parameters. So I think we've pretty much taken apart the theory and we've offered a viable additional theory as an explanation for that. And so, as I mentioned, you can get more information from the paper that I had at the beginning of this talk, or you can turn to our own webpage, as Dr. Thomas already mentioned, at icr.org and tune into any of these podcasts and get more additional information. So I know we covered a lot of information in a short amount of time, but thank you very much for enabling me to go through this and hopefully explain it for all of our listeners.
Dr. G, that was fantastic. That is real science right there. Your podcasts that are put out on the ICR YouTube channel are excellent. I really enjoy those. So great work to your team. And what I kept thinking was, when you start with a biblical worldview, you can make these types of accurate predictions on what to expect in the biological world. And so excellent job, Dr. G. That completes our second presentation for tonight's Science Confirms the Bible seminar.